In the frozen darkness far beneath the surface of Antarctica, a tiny community made its home. These were not penguins or seals, nor were they even fish. They were microorganisms so small and invisible to the eye that most people might not know they existed. To them, though, this under-ice world was everything. It was cold and vast, silent and isolated. From their perspective, it was the harshest place imaginable. It had always been that way, and they had learned to thrive within its relentless grip of cold and pressure. This community lived beneath a layer of ice called the Ross Ice Shelf, one of the largest ice shelves in Antarctica. Here, sunlight never penetrated. Day and night meant nothing. The temperature hovered at just above freezing for water, though, for these microbes. Even that felt like warmth, thanks to small pockets of geothermal activity. These natural hot spots came from the Earth's crust, deep underneath, and they provided enough heat to keep pockets of liquid water open. Within these water pockets, the microorganisms had everything they needed. Water, food, in the form of chemicals and minerals, and a relatively steady temperature. But outside these pockets, it was harsh ice that crushed with high pressure, total darkness, and a constant silence so deep it seemed alive. To live here, these microbes had evolved incredible ways to survive. With no sun to power photosynthesis, they needed another source of energy. They found it in chemicals dissolved in the water-bearing elements like iron, sulfur, and nitrogen. By breaking these chemicals apart in a process called chemolithotrophy, they created energy. It was a slow way to live, but reliable, and it meant life in this frozen world continued very slowly. But it did. For millions of years, this had been enough. Over time, these organisms adapted even more. Some grew special enzymes that worked better in cold. Others developed thicker cell membranes to withstand pressure. Some even produced tiny amounts of protective proteins acting like natural antifreeze, keeping their insides from freezing solid. Each microbe carved out its own niche, some near the warm vents, others in narrower cracks where minerals gathered, others near water pockets where pressure was balanced. Together they formed a small but diverse ecosystem, full of life, so tiny, most people would never see it. In a sense, this frozen underworld was a perfect laboratory for studying survival. With no predators, no competition from large organisms, and no human disturbance, it operated on its own terms. Food was scarce, but available. Temperature was constant, but low. Light was absent, but life found other ways to power itself. For the microbes, this was normal. This was home. Life rhythms meant growth and division, microbes splitting into two, slowly, over days, or maybe weeks. Every so often, a population would have a glut of nutrients from a fresh pulse of minerals brought by shifting ice or geothermal activity. Then they'd multiply slightly for a while, until food ran down again. Between pulses, they survived. It was a slow, steady existence. From an outsider's view, this was boring. But it wasn't. It was fascinating in its own way. These organisms painted a picture of resilience. It showed that life did not need sun or air as we know it. All it needed was a stable energy source, water, and time. Pressure and cold, instead of being obstacles, became conditions to which organisms adapted. These microbes rewrote what we thought of as the limit of life. Stillness was the setting for this world's elements, punctuated only by the occasional hiss or crack of shifting ice, far above, far beyond anything these microbes could even sense. To them, their condition was absolute. There was no warmer place, no more comfortable environment. They were adapted to the idea that their world was the worst there could be. This certainty shaped every aspect of how they lived. Their metabolisms were slow because reactions happened slowly at low temperatures. Their reproductions were rare. Their survival instincts were based entirely on chemical detection, not light or sound. They had no idea of earthquakes, no concept of time beyond the chemical cycles they rode on, no idea of animals or humans or life any differently. Their universe was dreary but dependable, predictable, secure. For countless generations, no change had come that mattered. And yet, as is often the case with life, the static state could not last forever. Though they could not perceive it, big changes were happening far above. The ice moved, the Earth's crust shifted, tiny cracks formed. These microbes might feel changes in temperature or pressure, but those were part of their world. They didn't worry. That was just Earth doing what Earth does. From this microscopic world, this was all life would ever be. Then, unbeknownst to them, even more dramatic change was about to come. Because somewhere, far away, humans prepared a mission, a small probe designed to burrow through the ice shelf to search for life. It would take months of travel. It would pierce the ice. It would enter the cracks and fissures, carrying warm fluid, recording equipment, sterilized steel, and eventually it would arrive in the world of these microbes without warning. Unannounced. For now, though in this part of the story, it had not happened yet. The microorganisms carried on as they always had. They consumed chemicals. They survived. They didn't know anything was coming. They didn't know that their steady, safe world would soon be altered in ways they could not imagine, because in their dark, frozen world, where the only light was chemical energy. That was how life went on quietly, slowly, and far from any human eye. And that is exactly how they wanted it to stay. Far above the dark, frozen world where the microorganisms lived, a team of human scientists was working on something big. They were curious about life in the most extreme places on Earth. For many years, people had wondered, could anything live deep under the Antarctic ice, where it's cold, dark, and full of pressure? It seemed impossible, but scientists had a feeling that life might exist down there, and they wanted to find it. So, they began planning. This wasn't an easy task. Reaching a world under almost a kilometer of solid ice would take years of research, hard work, and the latest technology. The ice was thick, the conditions were harsh, and they had to be very careful not to damage anything or bring harmful things into that untouched environment. Still, their desire to know more was stronger than the challenges. They designed a special machine, a long, tube-like probe that could melt its way down through the ice using hot water. It was equipped with cameras, lights, and tools to collect samples. They made sure everything was as clean as possible so that they wouldn't bring bacteria or chemicals from the surface into the hidden world below. They called the machine a clean probe. It had to be clean, because this mission was not just about finding life, it was about not harming it. While the scientists worked, deep under the ice, the tiny lifeforms continued their peaceful existence. They had no idea that something was coming. Their world was still, as always, cold and dark. The water around them was silent. The only changes they ever noticed were slow and small, perhaps a slight change in mineral levels, or a new crack forming in the ice. These things were normal to them. Then, very slowly, something new began to happen. Far above, the clean probe was finally launched. It began melting its way down through the ice, slowly making progress. To the scientists, it was a moment of excitement. They watched their instruments carefully, measuring the temperature, pressure, and movement. They couldn't wait to see what they might discover. Every meter the probe went down brought them closer to the unknown, but to the tiny creatures deep below, something was changing in their world. At first, it was just a faint warmth, the heat they had never felt before. It was different from the gentle warmth from the geothermal vents they knew. This heat was sharper, moving downward in a slow but steady way. Some of the microbes noticed it. Their chemical sensors picked up slight changes in water temperature, but they didn't know what to think. Was it just another shift in the Earth's crust? Soon, more changes followed. The pressure in their environment began to shift slightly. There were new movements in the water, teeny vibrations, like soft waves that came from above. These were the early signs of something big, but the microorganisms couldn't understand them. They had no idea that something completely foreign was approaching. Their world had always been quiet. Now, something was disturbing it. As the probe got closer, light began to shine into places where no light had ever been before. The probe had strong lamps attached to it. When they turned on, they lit up the water in bright beams, cutting through the darkness. To the microbes, who had lived for millions of years in pure darkness, this light was overwhelming. Many of them could not even sense light, but for the few who had some light-sensitive structures, an ancient leftover from a distant ancestor, the sudden brightness was like a bolt of energy. It was both fascinating and frightening. Along with the light came new chemicals. Though the probe had been cleaned carefully, nothing is ever perfectly pure. Tiny amounts of human-made materials entered the water: grease, oil molecules, bits of plastic, and metal. For the human scientists, these amounts were too small to matter. But for the microorganisms, these new substances were completely alien. Their bodies had no idea how to react. Some tried to absorb the new materials, mistaking them for minerals. Others avoided them. A few simply shut down and died. The probe continued to descend. It sent out sounds, high-pitched sonar pings and mechanical vibrations as it scanned the environment. These sounds traveled far in the still water. To the microbes, these were like strange songs or earthquakes. They had never heard anything like them before. Their world, once calm and slow, was now full of unknown sensations. Some microbes reacted by releasing stress chemicals. Others started forming protective layers, and some just floated, confused. When the probe finally reached the main water cavity where many microbes lived, it caused a gentle rush of bubbles and warm water. This sudden arrival felt like a storm to the microorganisms. The probe's lights scanned the area, and its cameras recorded every corner. It took water samples, sucked up a few of the tiny creatures, and measured the temperature, pressure, and mineral content. To the human scientists above, it was a moment of victory. They saw their first images of the under-ice world. They saw strange patterns in the mud, odd colors in the minerals, and tiny moving dot signs of life. Their hearts filled with joy and amazement. "We found it," they said. "There is life under the ice." But down below, that same moment felt very different. It was like an earthquake mixed with a blinding flash of light and an invisible chemical spill. For the first time in their long history, the microbes faced something they could not adapt to quickly. Their defenses were too slow. Their world was being changed faster than evolution could keep up. Still, some of them began trying. A few species started producing thicker cell walls. Others grouped together, hoping safety could be found in numbers. Some microbes hid in narrow cracks in the ice, escaping the probe's reach. In their own tiny ways, they began to fight back, not with weapons, but with the oldest tools of life: adaptation, resistance, survival. Meanwhile, the humans were celebrating. They published papers, gave interviews, and dreamed of more missions. They spoke about life in extreme environments, about the possibilities of life on other planets like Europa or if life could survive under Earth's ice, maybe it could exist elsewhere too. The discovery opened many doors. But few people stopped to wonder how the tiny life forms might feel. To the microorganisms, humans were giant intruders. Their arrival brought brightness where there was darkness, heat where there was cold, noise where there was silence. It brought strange materials in sudden change. The microbes had never seen anything like it. Their world, which they believed to be the harshest place possible, had just become even harsher, not because of nature, but because of something new: humanity. Even after the probe left, the environment was different. Some microbes had died, others had changed. The water carried new traces of chemicals. The balance of minerals had shifted. The ancient stillness was gone. In its place was something unpredictable and wild. To the microbes, the world was no longer just ice, pressure, and darkness. Now, there was something else, something they didn't understand, something that could return. But they survived. In their own tiny, ancient way, they endured. They had lived through countless shifts in Earth's history: ice ages, volcanic events, even meteor strikes. Now, they had lived through their first contact with humans, and though they didn't know it, that moment would change them forever. The scientists above had taken only a small sample, recorded only a few hours of footage, but they had opened a door. Now, nothing would be the same. And deep beneath the ice, in the cold and pressure, life went on. Slower than before, perhaps, a little more wary, but still alive, because that is what life does. It adapts even in the face of something it cannot understand. When the probe entered the hidden world beneath the ice, it did not arrive quietly. To the humans, it was just a machine doing its job, melting ice, collecting samples, and sending back pictures. But to the tiny creatures living in the cold water, it was something completely new, something far beyond anything they had ever experienced. The probe was a giant to them. It was made of metal and plastic, strange materials that had never touched their world before. It moved with force, pushed warm water out of its sides, and shined lights that cut through the darkness like lightning. For millions of years, these microorganisms had only known slow change, silence, and darkness. Now, all that was broken. The lights from the probe reached every corner of their watery home. The light was bright, white, and unnatural. It was not like sunlight, something these creatures had never seen anyway. It was artificial and steady, with no rise or fall. Some microbes had no way to even notice the light, but those that could detect changes in their environment felt a strange energy in the water. The light confused them. It made their world feel hot and exposed. The movement of the probe also disturbed everything. As it moved, it stirred up the mud at the bottom of the lake, sending tiny particles spinning through the water. These particles blocked chemical signals and clogged the feeding systems of some microorganisms. For a while, everything felt chaotic. Microbes that were once settled and safe were now floating, lost, or buried. More than just physical disruption, the probe brought new elements into the ecosystem. No matter how clean the probe was, it wasn't perfect. It released small traces of oils, metals, and even microscopic fibers from human materials. These were completely foreign to the native microbes. Their bodies had no idea how to process these things. Some microbes tried to eat the new materials, thinking they were minerals, but when they absorbed them, their internal systems broke down. Others were poisoned slowly. A few, more adaptable microbes started reacting in different ways. Some formed hard shells, some grouped together into sticky blobs as a way of protection. Their instincts told them something dangerous had arrived. The camera and sampling equipment added to the problem. The probe sucked in water samples, pulling microorganisms with it. The microbes had never experienced anything like this force. They were swept into tiny tubes, trapped in dark spaces, and pulled up toward the surface faster than they could react. Some died instantly. Others were frozen in the sample containers. Those that survived found themselves far from home, their delicate chemical balance ruined. Even the ones left behind felt the loss. They depended on each other to maintain balance in the ecosystem. Now, that balance was broken. The microorganisms, in their simple way, understood that their world had changed. Their reactions were not based on fear or emotion, but survival. Life is always trying to continue, and these creatures were no different. They began to change. The ones closest to the probe began building defenses. They thickened their cell walls. Some became resistant to the new chemicals. Others stopped moving completely, entering a kind of sleep state to wait until the danger passed. Some microbes responded with speed. Though they were tiny and slow compared to animals or even plants, their world moved at a different pace. In just a few hours or days, they could reproduce and pass on new traits. Microbes that survived the first disturbance began to multiply. Their offspring had slightly stronger membranes, better chemical sensors, or slower metabolisms to avoid stress. These small changes gave them a better chance of survival. And then, something remarkable happened. Some microbes began to interact with the materials left behind by the probe. A few could break down plastic fibers or use metal ions in new ways. This was not something they had done before, but evolution works in strange ways. The most flexible organisms tried everything they could to survive. A new kind of behavior was starting to appear, one shaped not by the old rules of their world, but by the influence of the humans who had entered it. To the human scientists watching from above, these changes were invisible. They were focused on their data images of strange underwater structures, measurements of chemical levels, and samples full of tiny life. They were amazed to find movement, to see living cells under their microscopes. They talked about how this proved life could survive in extreme places. They discussed possibilities for life on other planets, like Europa or Enceladus, where oceans might exist. They had touched something ancient and alive, and they were proud. But they did not fully understand what their presence meant to the world they had entered. The microorganisms had never known predators or invaders. They had evolved only in response to natural conditions: heat from the earth, chemicals in the water, the pressure of the ice above. Now, for the first time, they were adapting to something artificial. The humans, with their machines and curiosity, had become the first threat these tiny creatures had ever known. Still, the microorganisms did what life always does. They kept going. Some formed colonies that stuck tightly to the ice walls, far from where the probe could reach. These colonies created protective coatings, biofilms that made it harder for foreign materials to enter. Others moved closer to the geothermal vents, where the water was more stable and the probe's influence was weaker. A few even remained curious, testing the new chemicals, trying to use them as food or building blocks. All this happened without thoughts, without plans. These life forms had no brains or emotions. They responded with biology alone. But their actions told a story of struggle, of damage, of change, and of survival. In the weeks that followed, the probe was removed. The mission was complete. Scientists reviewed their findings, published research papers, and planned future missions. They saw success. They celebrated their discovery. But the story beneath the ice was not over. The probe had left behind more than it took. Its heat had melted new paths in the ice. Its chemicals had changed the water. Its presence had started a chain reaction. The world beneath the Ross ice shelf had been silent for millions of years. Now, it was awake in a new way. Life there had been changed not by nature, but by contact with a completely different kind of life. Some microorganisms had died, unable to handle the new reality, but others had survived. They had changed. And with those changes came new possibilities. They no longer believed their world was the harshest. They had learned that something even more powerful could exist: giants from above, with tools and heat and light, who could enter their world in a single moment and shift everything. To the microbes, these giants were gods or monsters. But either way, they were real. And now that they had come once, they could come again. So the microorganisms adapted. They prepared, not with weapons or buildings, but with changes to their cells, to their chemistry, to their patterns of life. They evolved. Because even in the coldest, darkest, most forgotten place on Earth, life never stops changing. Life never gives up. And though they were small, nearly invisible, and silent, the microorganisms beneath the ice were ready for whatever came next. Below the thick, frozen ice sheet of Antarctica, the tiny life forms called microorganisms were learning to live in a new world. For millions of years, they had lived in complete silence and darkness, untouched by anything from the surface. But all of that changed when the humans sent their metal probe down through the ice. That probe brought light, heat, and strange chemicals. It disturbed their world in ways they had never seen before. In the beginning, the microorganisms didn't know what was happening. They had no eyes to see the light, no ears to hear the noise, and no voice to share their fears. But they could feel the heat. They could sense the strange currents in the water. They could detect new chemicals in their environment. And what they felt was danger. Many microbes died. Some were swept away by the machine's suction. Others were poisoned by new substances. Their carefully balanced home was broken, and they had no time to prepare. But life, even in the harshest places, always tries to continue. The survivors began to change, and slowly, their tiny world began to evolve. At first, the change was small. A few microbes began forming thicker shells around their bodies. These shells helped them survive the toxic chemicals. Others developed sticky outer layers to cling to surfaces better, so they wouldn't be swept away. Some even stopped moving completely and entered a deep sleep, waiting for the danger to pass. But the danger didn't go away. Even after the probe left, the environment remained different. The water was slightly warmer in some areas. New minerals, and chemicals floated in it. The sediments had been stirred. Places that were once safe were now full of shifting particles. Food sources were gone. Chemical signals, which the microbes used to communicate and organize themselves, were now hard to read. It was as if a massive storm had hit their peaceful world, and now everything was mixed up. Some microbes didn't survive the storm. Systems were too delicate. Their food too specific. They disappeared quietly. But others stronger or luckier found new ways to live. They started to experiment with the new materials around them. One type of microbe began to use the strange plastic-like fibers the probe had left behind. At first, these fibers confused them, but then they started to attach to them. Later, they even started breaking them down, not for food, but to use as shelter. This behavior had never been seen before in their world. In another part of the lake, near where the probe had passed, the heat it left behind created new currents. These gentle flows brought minerals from deep in the lake to the upper parts, where no microbe had ever lived before. Some brave microbes floated into these new areas. The temperature was different, the water was clearer, but there was food strange. Food, but food, nonetheless. These explorers started colonies in areas that had been empty before. Their children inherited traits that helped them survive the new conditions. Stronger membranes, faster reproduction, new enzyme functions. Soon, patterns started forming. Microbes that could survive the new heat grouped together. They formed networks, sharing resources and shelter. They developed what scientists call biofilms, layers of slimy protection, where many microbes live together. These biofilms were stronger than anything seen before in that ecosystem. They grew on rock, on ice, and even on tiny particles. Floating in the water, some microbes began changing their feeding habits. They had once lived on simple chemicals produced by the heat of the earth or the minerals in the rock. Now, they began to use the tiny traces of human chemicals, oils, cleaning agents, bits of rubber, and metal as part of their diet. At first, these substances had seemed like poisons, but now, they were becoming part of the ecosystem. Microbes were evolving to not just tolerate these substances, but to need them. This was the beginning of a big transformation. A few microbes even developed chemical defenses. They produced compounds that made it hard for other microbes to take their space or eat their food. These chemicals spread slowly in the water. In some areas, they made it impossible for old microbial species to survive. Only the new, adapted forms could live there. In this way, the old world was pushed aside, and a new one began to take over. The most shocking change was seen in the microbes that had come in direct contact with the probe. Some scientists later discovered that these microbes had changed their DNA, not in a planned way. Of course, they had no brains or will, but by accident. Stress can make DNA break or copy itself wrong. Normally, this kills the microbe. But sometimes, it creates a new version, one that can survive better. These mutated microbes were spreading. They looked different under the microscope. They acted differently too. Faster, harder, more aggressive. This new behavior shocked the scientists when they returned later to study the same lake. They expected to find the same peaceful, quiet life they had seen before. But now, the microorganisms were more active. Their colonies were larger and more complex. Some species had completely disappeared. Others had become dominant. And some were completely new, never seen before in that environment. The scientists were amazed. They had never expected life to change so quickly in such a cold, closed place. But they also felt uneasy. They realized their small visit had changed everything. Just one mission had caused the whole system to evolve. What would happen if they came again? Would the microbes keep changing? Would they become something dangerous or something useful? One thing was clear. The world under the ice was no longer the same. Meanwhile, the microbes continued their slow, silent transformation. They did not know they were being studied. They did not know they had changed. They simply did what life does: survive, adapt, reproduce. And in doing so, they became stronger. The new world was no longer just dark and cold. It had warmth, movement, new resources. It was richer and more dangerous. Some colonies became very complex. Microbes lived in layers, each group doing a different job. Some made energy, others broke down chemicals. Some even acted as guards, blocking harmful substances from entering. These communities looked like tiny cities, organized and self-sufficient. This was something never seen before in that lake. And it didn't stop there, because life under the ice had begun to change. It also started to connect. The water carried tiny signals, chemicals used by microbes to talk to each other. These signals traveled farther now, reaching places that were once isolated. Colonies started reacting to signals from far away. One group might start growing faster when it sensed another group nearby. This kind of chemical communication was rare in such a cold, isolated place. Now, it was common. It meant the lake was no longer just a collection of small life forms. It was becoming a single, connected ecosystem. The arrival of the humans had made this happen. Without meaning to, they had pushed evolution forward. They had turned a stable, ancient world into a fast-changing one. Some scientists started calling it a second beginning for life under the ice, a rebirth. But not everyone saw it as a good thing. Some experts warned that the changes could go too far. What if the microbes developed traits that made them dangerous if brought to the surface? What if they became resistant to all known cleaning methods? What if, by disturbing this peaceful world, humans had created a new threat? Still, others believed it was a gift, a chance to study evolution in real time, a way to discover new medicines, enzymes, or technologies from life forms that had once been untouched by modern science. To them, the under-ice world was a treasure. Chest now opened. One thing everyone agreed on: the microorganisms were not the same anymore. They had changed, grown, learned. They had faced a great disturbance and survived. For the microbes, the story was simple. Their world had changed, and they had adapted. That is the rule of life. They did not remember what it was like before. They only knew how to survive now, but for the humans, the story was just beginning. They now faced a choice. Would they protect this changed world? Would they learn from it, or exploit it? Would they return and disturb it again? Or would they let it grow on its own? The tiny world beneath the ice was quiet again, but it was no longer still. Inside its silence was movement, progress, and life in new forms. And it all started when the humans came. Deep beneath the ice, the world was quiet again, but it had changed forever. For eons, the microorganisms under the Ross ice shelf had lived in a steady, unchanging realm of darkness, pressure, and slow chemical energy. Then humans arrived there. Probe pierced the ice, brought heat, light, noise, new chemicals, and a sudden rush of disruption. We watched what happened in part 3 and 4, how the microbes reacted as living things always do, by trying to survive, adapt, and change. Now, in this final part, we explore the consequences of that moment and how it transformed both worlds, the tiny under-ice ecosystem and humanity's understanding of life and responsibility. After the probe came and went, the human scientists on the surface returned to their daily routines. They analyzed their findings in labs, wrote scientific papers, and prepared presentations. They marveled at their achievement, discovering a living world hidden beneath a mile of ice. Their images showed strange microbial mats, swirling chemical plumes, and living cells glowing under powerful microscopes. They spoke of future missions to drill deeper, to explore more hidden lakes, and to search for life on icy moons beyond Earth. To them, this was proof that life can thrive in extreme environments on Earth and, possibly, beyond. It was a scientific triumph, but in the silence under the ice, life had changed. That change started small: a few dead cells, disrupted chemical signals, altered mineral patterns, and traces of human substances in the water. But small changes can lead to big outcomes, especially when life is involved. Over weeks, then months, the ecosystem began showing signs of transformation. Some microbial colonies had died out. They had been near the probe's sampling area or sensitive to the new substances. Where they once formed thick mats on rock or ice, there were now empty spaces, ghost colonies. But those spaces didn't stay empty long. Other, hardier microbes pushed in. They had survived the initial disturbance, perhaps adapted to the changed environment. They multiplied quickly, taking advantage of the new conditions. One key change was the presence of human-made compounds. The probe had left tiny amounts of lubricants, mild disinfectants, and plasticizers in the water. To most of the microbes, these were toxic. Their enzymes didn't recognize these substances, so they caused stress or death. Yet, within this harsh new soup, a few microbes thrived. They may have had enzymes with broad specificity, able to break down or tolerate the foreign chemicals. Those survivors reproduced, passing their resilience on. Soon, communities of microbes that could metabolize these compounds began to form. At the same time, the thermal patterns in the ice had shifted. The probe's hot melt created a path of slightly warmer water. Microbes near that channel found a new kind of home. They could live where the ice was thinner, the pressure a little lower, and the nutrients more concentrated from melting minerals. Their metabolisms always slow sped up just enough to allow faster growth. These warmer niches became microparadises, crowded with life, compared to the frozen stillness that had existed before. Life adapted to this heat. Some species even evolved small molecular changes to withstand the slightly higher temperature, and to resist the chemical pollutants. Another effect of the probe was mechanical disturbance. It stirred up sediments and minerals. Previously, most microbes lived in stable microhabitats, rock cracks, or tiny crevices in sediments. Now those sediments drifted, reorganized, and redepleted. That meant many habitats were destroyed but new ones formed. Sediment clouds carried minerals like iron, sulfur, magnesium, and silica. When they settled, they created fertile new grounds for certain microbes. These microbes prospered, forming new colonies in places untouched before. Within months, the under-ice ecosystem began to look very different from the one humans had glimpsed. Areas once smooth now had bright patches of microbial blooms. Some colonies glowed under specific lights when humans viewed sample signs of biochemical reactions. With the foreign chemicals, tiny, filament-like structures reached out, perhaps to capture nutrients or defend space. The microbes had created new biofilms, protective layers thicker than before. In microscopic images, these biofilms had multiple layers of cells bound together with secreted polymer, signs of increasing complexity. The probe had awakened the ecosystem. For millennia, it moved in near-perfect balance. Chemicals, energy, pressure, life. Now, the balance had shifted. Some microbes died. Some mutated. Some new species, or stray survivors from previous migrations under the ice, found the new conditions ideal and popped up. Genetic diversity increased as different strains multiplied. In effect, the probe and its disturbance had acted like a sudden attack, lethal to some, transformative to others. It accelerated evolution. Meanwhile, back on the surface, the scientists were celebrating. They presented their findings at conferences. They showed captivating images of ice water chimneys, microbial communities, and rare enzyme structures. They talked about the implications for astrobiology, for industrial enzymes, for our understanding of life's flexibility. However, few paused to reflect on whether their footprint under the ice would matter in the long run. They had stirred a world and left. It was out of sight, out of mind. Their transcripts included careful sections on sterilization protocols and minimal contamination, yet the results showed initial contamination had been unavoidable. They conceded that even tiny amounts could cause large shifts in such a closed system. Some scientists advocated slowing down. They urged more observation before further interference. They argued that ongoing sampling should be intrusive, slower, and limited in frequency. A few even proposed creating a sounding station, a permanently installed device to monitor changes over time without further disturbance. Others, excited by the discovery's novelty, pushed for faster, more frequent missions, deeper drilling, and aggressive sampling to unlock secrets of ancient ice, chemical cycles, and potential biotech applications. Their debate echoed earlier conflicts about scientific exploration. How much to take? How much to leave? It raised ethical questions. Was it right to expose a pristine ecosystem to even minimal contamination? Who would speak for the microbes? For centuries, humans have explored untamed frontiers in forests, deep-sea vents, deserts, and each time new species discovered were followed by disruption. Here, on Earth's final frontier, the under-ice biosphere, the same pattern repeated. Yet, unlike surface ecosystems, these microbes had no guardian. They could not protest. They could only adapt if they could. Their tiny genomes would record changes, resistance genes, new metabolic pathways, stress response proteins. Over generations, their descendants would be different. It would be impossible to undo. We had changed them forever. Over a year passed. Remote monitoring showed the warmed channel slowly refreezing. Sediment drifts settled. Chemical traces from the probe diluted below detection. Yet, where once only cold-tolerant microbes thrived, new communities remained where the disturbance had occurred. In one region, a thick microbial mat covered a rock face. Its structure, unlike anything found in surrounding areas. Genetically, it looked like a hybrid, part original cold-adapted microbes, part newcomers. Functional tests showed it could tolerate chemicals humans introduced, produce enzymes that could break down plastics, and even generate gases humans hadn't expected, small amounts of hydrogen sulfide or methane. These were chemical signs of new life cycles emerging under the ice. In a way, the disturbance had created something novel, a human-influenced biome. It was still dark and cold in most parts, but it had a hotspot, a place shaped by human presence. And that presence, though brief, had lasting effects. The ecosystem still existed, but it had changed, evolved, adapted. Life had continued, but on new terms. On the human side, researchers realized what they had done. While they had discovered life, they had also reshaped it. They had altered evolutionary paths. They had perhaps undone millennia of isolation. What would this mean for future missions? Should they accept the change and continue, or give the under-ice world time to recover? Should they leave it alone, or return in a way engineered to minimize impact? These questions weighed heavily on ethics committees, government agencies, and international bodies. Antarctica was governed by treaties about science and conservation. Now the debate touched even deeper ecosystems far removed from any human presence. More broadly, the discovery and its consequences sparked public interest. Documentaries asked, can we explore responsibly? News headlines asked if humans had the right to disrupt untouched worlds on Earth or beyond. Schools taught lessons. Life can exist without sunlight, but even small disruptions can echo through an ecosystem. For many, this under-ice mission became a metaphor. We are explorers, but we must also be guardians. Under the ice, life continued. Microbes adapted, evolved, and thrived. Their world was not as harsh as they once thought. It now included foreign energy, strange chemicals, and unpredictable disturbances. But it also had new niches, new food sources, new evolutionary opportunities. The cold darkness had been shaken, but now it teemed with life in hidden ways. The original aim had been scientific discovery, prove life existed. That had been achieved. But the legacy was deeper, a lesson in humility and responsibility. Even the most isolated place on Earth could not escape human touch once we reach it. The existential question. Can we learn to tread softly, to explore without forever changing? Deep beneath the Ross ice shelf, life went on. It changed and in change. It adapted. The microorganisms, once certain of their harsh world, had learned something new, that the greatest threats and the greatest opportunities can come from the tiniest spark. And from that spark, life had renewed itself in ways unexpected, resilient, and remarkable.